predicted. Yeah, somebody will show up on the hour. I just said. Okay. All right. Today we got a lot of ground to cover, and I'm going to see if this will be a first. If I can get through the Legionella as well, I'm going to try real hard. Usually I spend about about a million words a second in the last few minutes getting through the Legionella. Okay. Now, we're going to cover today the Mycobacteria and the Corynebacteria, and they're given a different color because it's got nothing to do with Legionella. These are acid-fast and related, and I did say acid-fast and related, and I didn't say acid-fast. And when we're going to go through the lecture, you'll see what I mean about these organisms. Okay, we've covered a little bit of this in one of the earlier lectures, but we're going to get some detail. We're also going to mention the Nocardia as in this lecture. The Nocardia, some of these organisms in this group, particularly the Nocardia and also Actinomyces, are very filamentous looking, and so much so that they look like fungi under the microscope. And so traditionally the Nocardia and Actinomyces have been covered in the mycology section. But I'm sure Dr. DeSalvo will mention this, but if he doesn't emphasize it for some reason, make sure that you know it, that these are not, the Nocardia and the Actinomyces are not fungi, they're bacteria. Okay? But that's just the way it's done. Mycologists deal with Nocardia, and so that's the way it is. Okay. Now the thing is, what I'm going to do here, all this stuff in yellow then refers to the Mycobacteria and the Corynebacteria, which are the two organisms I'm discussing today, acid-fast and related. I shall mention the Nocardia just in terms of the staining characteristics when I get there. And again, don't be confused, because in the handout it does mention that one of the Legionella micdadi, stains slightly acid-fast. And that was just a coincidence. It had to be in the same lecture. We just didn't plan on putting it there. So they've got nothing to do. The acid-fast staining of Legionella, they're totally different genetically. They've got nothing to do whatsoever with the Mycobacteria, Corynebacteria, and Nocardia. Okay? All right, so those are the different groups we're going to discuss. And unfortunately, we're going to see today, somebody asked me earlier on about the mechanism of the gram stain. You're going to see that not necessarily the staining, there are some staining characteristics reflect what's going on with the cell envelope structure, but it's not. But there's some little bit of complexity to it, and we'll get there when we get there. Okay. Now, unlike the Nocardia and some of the other organisms in this group, generally Mycobacterium tuberculosis looks like it's a rod-shaped organism. It does not stain with a gram stain. It is not. Sometimes people say, well, is it gram positive or is it gram negative? It is neither. It is neither gram negative nor gram positive, because nobody ever uses a gram stain on Mycobacteria. It's just not done. It's unheard of. Okay, they are acid. They're stained with an acid-fast stain. Okay, these are one of the few organisms that will stain with an acid-fast stain. And I'm not going to go into the details of the acid-fast stain, but it's the same sort of thing you do with a gram stain, except that the decolorizing agent has got acid in it, and these organisms, the stain stays put. They don't get decolorized, and that's what it means by acid-fast. The acid does not remove the stain. Okay, so these are specifically referred to as acid-fast bacteria, and they are when you say acid-fast, I never say always in these situations, but invariably you are talking about the Mycobacteria when you talk about acid-fast staining. Okay, the Mycobacteria, the classic disease that we're going to be covering here is tuberculosis, often referred to as TB, and in the older literature and old movies as consumption. Okay, and now in contrast, although Corynebacterium is a genetically related organism to the Mycobacteria, the pathologic mechanisms are totally and utterly different in Mycobacteria and Corynebacteria infections. And indeed, the Mycobacteria are the classic organism that people discuss when they're talking about a cell-mediated immune reaction and the tissue injury being immunopathologic in nature. In the case of Corynebacterium diphtherium, on the other hand, essentially is one real big deal in the pathogenesis, which is the exotoxin. And indeed, certain strains of Corynebacterium, in this room, some of you probably now have Corynebacterium diphtheriae in your throat, but you do not have diphtheria, because the exotoxin, the strains only produce diphtheria if they produce an exotoxin. And many strains of Corynebacteria and diphtheria do not make exotoxins. Dr. Mayer, I think some while back, mentioned to you about, and I'm going to say it again, that there's a phage coding for the toxin in Corynebacteria and diphtheria. If you have a strain that doesn't have the phage, it doesn't have the toxin, it doesn't cause disease. And the immunization in this instance, the reason we don't see diphtheria in this country is that everybody in the room has been immunized against diphtheria. Okay, so these are two totally different issues, and it's useful to compare and contrast them as you go on. You see how different an immunopathologic disease and how you deal with it versus one that's exotoxin mediated in nature. Okay, so M. tuberculosis is the major human disease among the Mycobacteria. Nowadays, we recognize that it is associated with AIDS. What does that mean, associated with AIDS? Well, let me explain what I mean by associated. Around 1975 or so, when I was doing my PhD, for some reason or other, I was actually, I'm not going into detail, I was planning to infect a rabbit colony, which I subsequently did, with Mycobacteria. And I consulted all the experts that I could find on Mycobacteria and went back into the literature, and there hadn't been a case of this is 1975 I'm talking about. There had not been a case of M. avium in humans that I could find documented going back to 1924. Okay? It was almost unheard of. The AIDS epidemic came along. It was new to the world stage. And all of a sudden we started to see M. avium infections. And in fact, about half the cases, I think it's around that number, of AIDS-associated TB is caused not by M. tuberculosis, but by M. avium. So if you see a case of M. avium in a patient, it doesn't absolutely mean that they have AIDS, but there's probably 99.999% that they do have AIDS. Because that's why there are a number of opportunities that just don't show up essentially anywhere except with AIDS. But among the bacteria, Mycobacterium avium is one that you just essentially did not see in a normal, healthy human being, but is a big deal in AIDS-associated tuberculosis, and I will cover that in more detail as we go along. Okay, the other new thing for the last 20, 30 years or so, is that unfortunately the Mycobacteria were bad enough to treat in the past, but there's been multiple drug resistance that's shown up, and it is somewhat of a moving target, sort of selecting antibiotics in this disease. And I said to you, I won't mention. There are thousands of antibiotics out there I won't mention, except in situations where it's very important. So I'm going to mention a little bit about antibiotics in this situation as we go along, for that reason. Okay, the M. avium intracellulare complex, M. avium, it's sometimes referred to as the MAC complex. I hate abbreviations, I've said this before, but it is referred to as the MAC complex. The reason it's referred to as the MAC complex is because there are a number of organisms, including M. intracellulare, which are almost impossible in the laboratory, or extremely difficult to distinguish from M. avium. Okay, so the thing is, okay, never mind. Okay, so that's the situation there. Okay, so sometimes referred to as the MAC complex, but usually as M. avium. And again, as I already said, about outside AIDS, you almost never see it. It is the major bacterial opportunistic in AIDS, and there is multiple drug resistance to M. avium, not surprisingly as well. And unfortunately, it doesn't always respond to the same drugs as M. tuberculosis responds to. So it's bad enough treating M. tuberculosis. It's even worse treating M. avium, particularly in an AIDS patient. Another organism is mentioned, more for historical reasons, is because M. bovis, Mycobacterium bovis, and its name, as its name suggests, affects cattle. And it was a common thing in cattle herds in this country. But cattle in this country are always tested with PPD. There's a skin test done for Mycobacterium bovis. And if you ever see an organism that's a cow that's infected with M. bovis, it's removed from the herd, and the other cattle will be culled as well. So M. bovis is nowadays almost not seen in this country. It was a milk disease in the past primarily, but you just don't see this in this country anymore. M. lepri is another one I want to mention, Mycobacterium lepri. This is actually just as important worldwide as M. tuberculosis. There are literally millions of cases of leprosy around the world still. It's extremely rare in the US. Of course, a traveler to this country could have leprosy. But in addition, and you don't need to put this in your notes, and it's certainly not in the handout, There are armadillos in Texas that are naturally infected with M. leprae. And occasionally you see a case of leprosy in this country coming from these infected armadillos. And actually armadillos are very important when it comes to the study of leprosy. And if I remember, I'll get to it when I hope in the right place. All right. So the other mycobacteria are referred to as atypicals. The term atypical is a bit vague because back in the 70s, there are a lot of changes have occurred now, and sometimes the names have still stuck around. Back in the 70s, the situation was if you saw M. tuberculosis, that was it. If you saw anything else, what the clinical microbiology lab would do was throw the plates away and consume it so it was a contaminant and not even worry about it. And that was a standard operating procedure. But since the situation with M. avium and AIDS has come along, people have changed their minds. And nowadays people consider any time you see a quote-unquote atypical that, it could be involved in a case of human disease. And what I mean by atypical, let's see if I've got it on the next slide or not. Okay. What I mean by atypical is that M. tuberculosis is a very slow-growing organism, and it takes 3 to 6 weeks to grow up in culture, as does M. avium. It also produces non-pigmented colonies. Some of the mycobacteria produce coloured colonies, in addition to which some of the mycobacteria grow quite fast in a day or two. M. smegmatis is an example. So the issue comes down to is that they're atypical. And what exactly does atypical mean? Is M. avium an atypical? Well, it was an atypical in 1976. Is it atypical in 2006? Who knows? It's so common in AIDS, it's not atypical anymore. But that's where the word atypical comes from. They're the mycobacteria which are not typical. They're the ones that you generally didn't worry about in the past, and now you do, because it's now recognized that some of these atypical mycobacteria are causing human disease. But it's not common. The big deal in this country is M. tuberculosis and M. avium. The second point out of this is you just don't disregard something just because it's not M. tuberculosis or M. avium. Transmission, tuberculosis. M. tuberculosis causes disease in healthy individuals. It also causes disease in AIDS patients. We're going to have a few slides on AIDS alone. M. tuberculosis is transmitted man-to-man in airborne droplets. The pathogenesis of tuberculosis, not surprisingly, since it does involve transmission in airborne droplets, the infection is in the lung. It is the primary place that it infects, and it's distributed within macrophages. However, the organism does spread to the other tissues, and it's not just a pneumonia. That is the primary thing you see. But there's often infection of other tissues. This is a facultative intracellular pathogen. That means that it really prefers to grow inside cells. And in this instance, you really don't generally see acid-fast bacteria outside cells, or very few. It inhibits phagosome-lysosome fusion, and it also resists lysosomal enzymes if fusion were to occur. Cell-mediated immunity, tuberculosis. Okay, that is the hallmark of the disease. Get that in your mind. Cell-mediated immunity. You know your immunology, you know tuberculosis. There's an infiltration of macrophages and lymphocytes, which of course is typical of cell-mediated immunity. There are granulomas formed, and sometimes, they're often in this disease given a particular name, which is a tubercle, which specifically refers to a granuloma in tuberculosis. And I know you've had some of this in pathology, so that will, bring that together will help. Again, we talk about tuberculosis. We're usually talking about cell-mediated immunity. Anything to do with tuberculosis, not any of it. The vast bulk of what we talk about is to do with cell-mediated immunity. There is a simple test for testing if somebody has immunity to M. tuberculosis. The key thing I said there was immunity. I didn't say that they have tuberculosis. I said it's testing for immunity to M. tuberculosis. Why is there a difference? The reason there's a difference is this, that M. tuberculosis, the vaccine stinks. Okay, it's as simple as that. But there have been a number of countries, England was one of them, India was another, that in the past used M. tuberculosis vaccines. In the US, the vaccine was never used. And we don't have time to debate today why that happened and what have you. But so the thing is that if you were to get a foreigner, like me, for example, I'm sure I've not been tested in recent, but I'm sure that I have a positive skin test for tuberculosis. I'm also absolutely sure I don't have tuberculosis. Okay, so the point is that just because the test comes up positive, it doesn't mean somebody has tuberculosis. A skin test, testing for delayed hypersensitivity against the organism. It simply means that they are immune. Now, in this country, the vast bulk of the time, if somebody does have a positive skin test, they probably do have tuberculosis. Okay? So that's the issue. There's more to it. You don't just do a skin test in tuberculosis. You do a skin test, you think, okay, now I better deal with this because this person might have tuberculosis. Then you do other things. Now, in the skin test, what happens, there are two different, there are preparations used in tuberculosis were injected into the skin, and there are various versions of this test, and I don't propose to go into the various variations. It's unnecessary. But tuberculin is referred to as tuberculin or protein purified derivative, PPD. These are the terms that are used. Tuberculin, because it's prepared from M. tuberculosis, and protein purified derivative, or PPD, because it's supposedly purified, but it isn't very pure. It's actually junk. But still, that was the way the term, it's a partially purified extract and precipitate of the proteins of M. tuberculosis, and that is a common term, PPD test or tuberculin test. Okay? All right, the positive skin test in tuberculosis, as I said, it indicates exposure to the organism. It does not necessarily indicate that there is an active disease going on. So it's a very, very useful test, but it doesn't mean that you just make a diagnosis based on the fact of, that there's a pneumonia there, and the fact that there's a positive skin test does not mean the person's got tuberculosis. Generally, in this situation, you would go ahead and do PCR oftentimes, or you would do culture, depending on how the lab is set up. But you certainly confirm that that M. tuberculosis, the organism, is there, not just that you have a positive skin test. I just, for the sake of completeness, mention the Mycobacteria as a siderophore, and I mention a thing called cord factor, which have been suggested as being pathogenesis factors in tuberculosis. But the truth be said, the primary thing in tuberculosis that we care about is outside of exams and, national boards and what have you, is cell-mediated immunity. That's the big deal in tuberculosis. All right. Laboratory diagnosis of M. tuberculosis or TB. It is acid-fast. Now, I'm going to show, I'll have one slide, I'll get there when I get there, that goes a little further than this, and we'll get them with it. It is acid-fast, and it is the primary situation where one uses the acid-fast stain. And it's detected in sputum or a spectorate that you cough up from within your lungs. That's what sputum is. And again, I know that I've said this in other situations, but it doesn't do any harm to repeat it in a different situation. Just because you look down a microscope, if you see the organism there, if you see an acid-fast organism, that person has very likely got tuberculosis. If you don't see any organism in an expectorate or sputum, it doesn't mean the person has not got tuberculosis, because it only works about 50% of the time. So it's a very useful test when it comes up positive. But you would always go ahead in tuberculosis and either do culture or PCR or both to basically confirm that you have an active infection. Okay, again, laboratory diagnosis, a little more. Culture, the organism, as I said, grows very slowly. It takes several weeks to grow this organism up. Why do we change it? Because the media got better over the years. It used to be 6 weeks. Apparently now it's about 3 weeks. So I'm not going to make a debate about that. Several weeks. The organism grows very slowly in the laboratory. It produces non-pigmented or white colonies. And this is a useful test because if you really want to know that you've got TB, the organism grows so slowly that it's nice to be able to do a rapid test once you've grown it up for niacin, because this organism produces niacin, and most of the other Mycobacteria don't produce niacin. So that's a test that's used for M. tuberculosis. But in this day and age, with the advent of very good PCR tests that can rapidly tell the difference between M. avium and M. tuberculosis and give very specific diagnosis, I don't know if anybody really does this anymore. But that is an older test for M. tuberculosis. And as I mentioned, I stressed in another lecture, PCR for rapid diagnosis. If you've got an infection with E. coli, and you can grow the organism up in 24 hours in the laboratory, It doesn't make much of a difference most of the time if you get a PCR result in 4 hours or 24 hours also, as it depends on the version of the test. But in the case of tuberculosis, it does matter that you can do things more quickly. Because, for example, a teacher in a school might have developed tuberculosis and will be in contact with the children for weeks, months at a time. And there are cases where it spreads from an adult to children or vice versa in this situation. So if you know somebody who's got tuberculosis, you want to get that person out the way and get them on treatment quickly. Now, once they've been given the drugs for a few days, they're not going to be spreading this thing around. Okay? But you need to know. And so that's why we need rapid diagnosis. We don't need rapid diagnosis in tuberculosis because of the fact that it's going to kill anybody. It takes years, tens of years, for this disease to progress to its final stages. So that's why we need to know quickly, because we don't want somebody spreading tuberculosis around. Okay? So it is important that we have a rapid diagnosis in tuberculosis. In the past, the regimens have been as long as a couple of years. Now they've been cut down in many cases to as little as 9 months, but that's a long time to be on an antibiotic, I think. And the reason you're on this antibiotic, in addition, there are other differences in this situation, because the organism grows very slowly. It can, in some instances, become dormant. It gets trapped inside these tubercles or granulomas and hidden away. And it can take a while before you can clear all of this organism out of your system. So that's for the treatment. That's why the treatment is so long. In the past, it would have been said you would use 3 antibiotics. The thing is, in a disease of this type, what happens is that because it's so long that it goes on, there's a possibility during a 9-month or 2-year period that you could indeed develop, the organism could develop a resistance while it's inside the person. But the chances are, mathematically, since mutation rates are quite low, it's like very, very hard to get a mutation to 2 different antibiotics over that time period. And as I say, they used to do 3 different antibiotics, and it's almost impossible to get a resistance developed to 3 different antibiotics in a single patient over a 9-month period. But anyway, the tendency seems to have been to cut back from 3 to 2 antibiotics, and there are others. And I'm not going to get into every antibiotic because I don't know those sort of things, and it's going to be different from patient to patient. But one of the standard things at the moment is to use rifampin and isoniazid in particular. Okay, that minimizes resistance. Vaccination. Physicians might say to you, well, we don't vaccinate, so who cares? But you do care. The argument that's made is this. Okay, in the United States, and I think the rest of the world has gone along with this now. In a situation where you have a very low incidence of tuberculosis, there are only about 20,000 total cases, let's say new cases, around about 20,000 cases at a time of tuberculosis among a population of like 260 million we've got at the moment. So it's a lot of cases, but it's not sort of huge. And so the thing is, to vaccinate 250 million people, as I've said before, is not something that you're going to take on lightly, particularly when the vaccine is rather poor, and that's been shown experimentally in clinical trials. So the situation is that in this country it's felt that the incidence is so low that it's basically not worth it to even bother to. Now, in other cases like India, it was debated because the incidence might be higher, and so it's been debated it might have some use, if not a great use, in cutting down the incidence of the organism in the population. But in this country it's felt that the skin test is very useful. And so if you vaccinate somebody against tuberculosis, they're going to be immune, but they're not going to have TB. And so if you vaccinate, it causes a problem because it basically eliminates the usefulness of the tuberculin test, one of the rapid screening tests for this disease. Okay, so that's the situation as it's felt in the United States, and I think most of the world, the whole world, most of the world feels the same way at the moment. It didn't 20 years ago, perhaps even 10 years ago. Because, for example, in England, they did vaccinate when I was a kid over there, but they've stopped the vaccination program now in England the last few years. Okay, so that's tuberculosis. Now we're going to talk about leprosy. And yeah, that's what leprosy can look like. It's a horrible, nasty, disfiguring disease. Why is it that we're looking at the skin here? Because the organism grows best at 33 degrees centigrade, not 37 degrees centigrade. And the skin is somewhat lower in temperature. And so this organism tends to infect the skin and the nerves. And that's why you will see this disfigurement. Well, there are other reasons you'll see the disfigurement, but certainly it tends to grow preferentially in the skin as opposed to spreading throughout the whole body. Leprosy is sometimes referred to as Hansen's disease, because when a patient hears they've got Hansen's disease, they probably won't know what it is. But if you tell them they've got leprosy, they might get very upset. So it is referred to as Hansen's disease, but leprosy is the most commonly used term. M. leprae is the causative agent of leprosy. And as we've shown, it causes disfigurement. It's rarely seen in the US. I mentioned when you could see it. It's very common in the third world still. There are millions of cases. Now, and again, it infects the skin at a low temperature. And this is another example of what happens in leprosy, where the fingers have fallen off. And there's nasty jokes that I've been guilty of making them myself about leprosy patients. I won't make one today. But the reality is true. The skin can come off, the fingers can fall off. It's a very nasty disease. There are two forms of leprosy that are described. And it seems a little strange when you first hear it, but it makes sense if you think about it. We actually do use the word tuberculoid in leprosy, talk about in leprosy. We don't use the word tuberculoid when we're talking about tuberculosis. Basically, there are two forms of leprosy. This organism potently can inhibit the immune system. So what you can find in some cases, it looks like tuberculosis. In tuberculosis, we have a very active immune response, and we don't get a lot of organisms generally in the tissues, with the exception of AIDS-associated tuberculosis. But in a regular tuberculosis situation in a healthy person, you see a low number of organisms in a tissue, in a skin biopsy. But you will see, because you've got active immunity, which is controlling the replication of the organism. Now, this organism is not all the time, but in many patients, it actually suppresses the immune response. It's not quite clear exactly what the mechanism is, but it can cut down the immune response. Now, if you get immunosuppression in this patient, what that means is the immune response is not doing very well, so the organism can grow better. So you'll see a lot of organisms in a skin sample. But now, of course, there can be anything in between. At the one end, you've got tuberculoid. At the other extreme, you've got the lepromatous, and of course, you can imagine anything could happen in between. Okay, so there's a spectrum of disease in leprosy, which you don't see in tuberculosis. Yeah, go ahead.
Is tuberculosis was kind of same as tuberculosis?
Well, histologically, yeah. It means tuberculosis-like, tuberculoid. That's the term that's used. Okay, production of M. leprae antigens and pathogenesis studies. Now, this organism is yet another example. In 2006, a disease that causes millions of cases around the world, it cannot be grown in the laboratory. Now, class usually bursts out laughing at this point, but honestly, it's true. The armadillo is very, very helpful to us, because it turns out originally that people had to produce vaccine candidates and other experimental studies in the mouse foot pad. Because it's very, obviously, as I said already, the skin tends to have a slightly lower temperature than the rest of the body. Well, mice are pretty small, and you can imagine a foot pad of a mouse is not that big. Armadillos, it turns out, have a lower body temperature, which is presumably why they naturally develop leprosy. And it's widely used by people who work with leprosy to actually study and come up with new drugs and vaccines. Okay, so the armadillo is very important in not just the fact that it occurs naturally in Texas, and you can see a few cases here and there of leprosy in this country, but also the fact that it's used by the drug industry. Now, there is something that equivalent to the PPD or tuberculin test in leprosy, and that's skin testing. Again, this is an acid-fast organism. This is still Mycobacteria we're talking about, not Nocardia, not Corynebacteria, not Actinomyces, etc., etc. So the acid-fast stain is also very useful in leprosy. And the clinical picture is sure useful. And you've seen what the clinical picture, of course they were severe cases I've shown you there. But the clinical features are, of course, very important in the diagnosis of leprosy. Now coming back to the atypicals. And I don't think anybody would try and trick you. It's really, it's too complicated to trick anybody. Okay? The issue is that the atypicals, again, refers to organisms that grow differently, They have different colors. They're different than M. tuberculosis. And is M. avium an atypical? Well, it was in 1976. Is it an atypical now? I think it's pretty typical. So, I'm just trying to understand what the word means, okay? That is a term that's used. There are other terms that are used similarly, talking about these rare cases of tuberculosis with an organism that's out of the normal, okay? All right, so you know the terminology now. All right, so the atypicals infect the immunocompromised host in general, and they're not transmitted in man-to-man, and that says healthy people, and that's the big deal. I mean, if you had a few AIDS patients who are infected with tuberculosis or M. avium, I suppose it is possible that it could spread from one to the other, because the immune system is very poor. But it's very difficult to spread tuberculosis from one person to another. So if you're it doesn't mean you shouldn't take precautions, but if you're treating a patient with tuberculosis, it's very unlikely that you would catch tuberculosis. Now, you would be monitored regularly with X-rays and not skin tests probably, because if you monitor a resident or a nurse constantly with this test, they're going to come up positive, and then you might as well not bother to do the test anymore. But, there's things you can do. It's very hard to catch this disease. Now, on the other hand, school kids is a different issue, because again, you're sitting in very close quarters, you're exposed to one another all the time, not just for a few minutes, but hours and hours every day. So that's, again, why you really want to monitor a patient and treat them quickly and get them out of the way so they're not infecting other people. But it is difficult to catch tuberculosis. Atypical mycobacterial diseases. I think Dr. Bryan will go into a lot more detail than I do. He likes these things. But I've just decided to call them tuberculosis-like or leprosy-like, because that's the category they fall into. Some of them will produce that when we defined already. And when I say leprosy-like, I'm talking about lepromatous leprosy-like, because tuberculoid leprosy looks just like tuberculosis. I should have said lepromatous leprosy there, but whatever. They histologically and otherwise look like tuberculosis or leprosy, okay? All right. I don't want to get into there's a lot of different mycobacterial species out there, and I just think it's just beyond the call of duty to memorize all these names. But you do need to know M. tuberculosis in particular and M. avium for the reasons that I have discussed. And I think it's a free world. Other faculty should do what they do, and if you really want to get more detail, pick up Murray and take a look at it. All right, Mycobacteria and AIDS. M. avium is much less virulent than M. tuberculosis, and that's obvious, because you essentially don't get it in a healthy person. You only get it in an immunocompromised patient, particularly an AIDS patient. It essentially does not infect healthy people, as I've said already. Now, in M. avium, what happens is when the T-cell count gets low, that's when the lower the T-cell count, the greater the chance that the patient would develop an M. avium infection. Now, you do see M. tuberculosis commonly in an AIDS patient. Why shouldn't you? M. tuberculosis is a much more pathogenic organism than M. avium. M. tuberculosis can cause disease in a healthy person, so why shouldn't it cause disease in an immunocompromised one? I mean, it's common sense, isn't it? So you can see M. avium and M. tuberculosis associated with AIDS, but if the person's got an M. tuberculosis infection, it tells you a lot about how to treat the patient, but it doesn't tell you anything about whether they've got AIDS or not. Okay, so the emphasis is M. avium associated with AIDS, not because it's the major disease. And tuberculosis associated with AIDS can be much worse. But it tells you something in addition, knowing whether you've got M. avium, because if you see M. tuberculosis, well, it could be anybody in the population could have M. tuberculosis. It does tell you, but it does make you think for a little bit and say, I better do some more tests here and see what's going on. Okay, and so M. tuberculosis infects healthy people. It also infects AIDS patients. But, as you would anticipate, it infects earlier on in the disease. And there's more likelihood of the disease being systemic in an AIDS patient with M. tuberculosis than in a regular person, because the immune response is not very good. And this applies, so clinically, when you're looking at AIDS-associated tuberculosis, whether it's M. avium or M. tuberculosis, systemic disease is more prominent versus pulmonary in AIDS than other situations. And the lesions, again, this word lepromatous is very important. They're often lepromatous. That is to say, there's a lot of organisms in a tissue because of the limited immune response. Okay? So you really should fix your minds on those terms, tuberculoid and lepromatous. Think about what the situations are, because they're very useful terms. And again, as I said right at the beginning of the lecture, we're emphasizing here the cell-mediated immunity when we talk about these diseases. But you can see it gets quite complicated. But if you really fix in your mind, well, okay, when do you see, when is it? What do you see in a strong immune response? What do you see in a weak immune response? When do you see it? What would you expect? It's pretty straightforward once you get that picture. Okay. All right. So antibiotic therapy is primarily selected for M. tuberculosis because it is the more virulent organism. But if M. avium is involved, other antibiotics are often included. And I've mentioned a few of them in the antibiotic lecture I gave. I mentioned ethambutol, and I mentioned about several of the antibiotics back in the antibiotic lecture, which are not used generally for other rifampin, for example, and not generally used for patients other than M. avium or M. tuberculosis patients. There are some strange terms that are used, and I've chosen not to use them because in this day and age, I don't believe anybody who works with Mycobacteria would use them. So I will give you the simpler version. These organisms can be pigmented or not. Now what you do need to know is the pigmentation can occur in the light or it can do in the dark. In other words, if I put an organism the colonies are not coloured, you might put it in an incubator or in a cupboard and get it out of the way. And as it's growing, pigmented colonies can develop. And those people who are experts, and I don't claim to be one, on the mycobacterial species and strains, this is helpful. But in this day and age, it's really probably not the way it's done. The other issue is I mentioned that whether they grow fast or slow is also useful. But the reason, in this day and age, we don't have to simply see is something pigmented or isn't it pigmented, is it slow or is it not slow. That's helpful. But there are very, very good tests out there, and there are actually 3 different groups of tests because people consider this so important. I've mentioned already fatty acid profiling in another lecture, which I distinguish from metabolic profiling. These are the long-chain fatty acids that are found in cell membranes. It's been particularly used in the case of speciating Mycobacteria, and it is technically complicated, and only reference labs would do it. Mycolic acid profiling is even worse. Because it's just technically very complicated. It's done on a high-performance liquid chromatograph, originally developed at the CDC. Nowadays, most people would almost certainly be looking at genetic markers involving PCR. There are very clear markers that can differentiate by PCR between M. avium and M. tuberculosis and pick up the infection directly in a body fluid or tissue. Okay, but all 3 of these things are used. That can be used for any organism, as I've mentioned before. But there are reasons that you use it in a particular case. In this case, it was because there were lousy tests and so many different species of Mycobacteria out there that with one test, you could actually look at all the species at the same time. And that's why these things were done. Now, this is something I've mentioned a few times. We didn't actually get there. We finally got there. Those of you who keep wondering why I keep talking about mycolic acids and acid-fast staining. All right, now this is the situation. The Mycobacteria have very long-chain mycolic acids. I remind you, these are branched fatty acids that are they're unique to the Mycobacteria structurally. They have very long fatty acids. We're talking about they can be 100 carbons in length. The mycolic acids in Mycobacteria tend to be extremely long. I think it was Nemo was talking about hydrophobicity and hydrophilicity. I think those words are very important. I use them a lot. In this instance, these are going to be these are very hydrophobic, and that's why it's so hard to decolorize with the acid-fast stain. So that's why the Mycobacteria are the most acid-fast among these organisms. Now, as we go down the list, the Nocardia, which you're going to be talking about with the fungi, but you know it isn't a fungus by now. The Nocardia have intermediate chain length fatty mycolic acids, so they're defined as weakly acid-fast. The Corynebacteria are genetically related to Nocardia and Mycobacteria, but they have the shortest chain length in these mycolic acids. And because they're short chain, they're much less hydrophobic, and so these organisms are not acid-fast. Now, in this instance, it's pretty. So I want to go so don't just look at a stain and say, well, that's always happens in this situation. These are genetically related organisms, but they have different mycolic acids and they stain differently. Corynebacteria are considered to be gram-positive organisms, as opposed so they're acid-fast, weakly acid-fast, or just referred to as gram-positive, because you do not do an acid-fast stain for Corynebacteria. I've used the term before, pleomorphic. Okay, we've dealt with tuberculosis now. We forget about cell-mediated immunity. Now we're going to deal with exotoxins. The Corynebacteria are gram-positive organisms. I've coloured them yellow here just to show that they're related to the Mycobacteria, but they're gram-positive organisms, as it says down here. They're strict aerobes. They're pleomorphic. You can see they're pleomorphic. There's one there that's got this bulge at the end, and no, they're not spore-forming in this instance, but it's got a bulge on the end. That one there is a straight rod. These are pleomorphic. Now, diphtheria in this country is almost unseen by physicians, but everybody's vaccinated. Just because it's not seen doesn't mean it's unimportant. This happened a few years ago, as I mentioned in an earlier lecture. In Russia, when things broke down a few years ago, it's better now, but when things broke down for a few years, diphtheria suddenly went from nowhere, it suddenly shot up over about a 5-year period. If you are all infected, or not all of you, many of you are infected with this Corynebacterium diphtheria. It's common in the human population, but it often doesn't have the phage, which codes for the toxin. But in addition, we're all vaccinated as young children. So we don't see diphtheria because the vaccine's great. I've so many times said the vaccine's not good, not good. Well, the vaccine's terrific. It works. Now, in diphtheria, we can see two aspects to the disease. First of all, it's an upper respiratory infection of the pharynx as opposed to the lower respiratory infection, the pneumonia we talked about in tuberculosis. This is an upper respiratory tract infection. And what can happen is you can kill you in two ways. The first way it can kill you is a pseudomembrane develops and it chokes you. That's a local reaction. The bacteria do not spread systemically. That does not mean that you don't get systemic disease. You sure do get systemic disease in diphtheria. Let's show you the local reaction first. And that would be a child that's basically choking to death from suffocation. Diphtheria toxin. Now, the organism does not spread. It's a local infection in the pharynx. So in this instance, what we mean is you might have, let's say, for argument's sake, let's say in a particular field in a microscope, there was half a dozen organisms. Well, maybe there might be hundreds of organisms, thousands of organisms in an active infection. So what we mean is that what's happening is the organism is really taking off and growing and producing a toxin in non-immune individuals. The toxin can spread, and it does cause systemic and fatal injury, and that's why everybody's vaccinated against it. Now, we talked about these ADP ribosylating enzymes before, and this is one among the four, but this is the primary one with the E. coli toxins we talked, Pseudomonas toxin that we talked about earlier. It's an ADP ribosylating enzyme, and ADP ribosylates elongation factor 2. What does that mean? No protein synthesis, cell death. That's what happens in diphtheria, death. Okay, it's a very nasty disease. Okay, treatment. You can use an antitoxin. In this instance, we know the toxin's pretty good because it's used as a vaccine. The antitoxin would help, and of course, antibiotic therapy. And we're all immunized against diphtheria as infants. The disease has vanished in the US, and without immunization, there's no doubt it will return. And if you're ever talking to families, people say, well, why am I doing this? Why am I having my kid vaccinated when the disease is not out there? Well, first of all, because the kid could get the disease. But more importantly, if you don't vaccinate the population, it may not be your kid that gets it, but somebody else might. And so vaccination is very important, and schools will often not allow children to come to school unless they've been vaccinated for very good reason. Okay, so the immunization is against the diphtheria toxin. It's referred to as a toxoid. And immunization is also against pertussis, that Dr. Gafar will discuss, and tetanus, that I've already discussed. An old test that's sometimes mentioned is the Schick skin test, a similar sort of thing that we do with tuberculosis. It's a different mechanism, and I don't think in this day and age it's really worth spending any time on it. You've heard of it, though. In this case, it's testing for basically, is there a toxin present? It's not testing for cell-mediated immunity. Dr. Mayo mentioned earlier, diphtheria toxin is coded on a bacteriophage. There's a so-called tox gene there. It is not synthesized in the presence of iron. An iron repressor complex forms which inhibits expression. So a phage, the presence of a phage coding for the diphtheria toxin is a big deal. If somebody's got Corynebacteria and diphtheria in their throat and they don't have the phage, it cannot cause diphtheria. Okay, now this organism, unfortunately, it does have some very unique characteristics. It's usually grown on what's called Loeffler's medium, or one of the media it's grown on. And you stain for, I mentioned about these storage granules in an earlier lecture, and these organisms particularly produce a lot of these polyphosphate storage granules. And what happens is, the term that's used here is metachromatic, more than one color. You often see the polyphosphate granules will stain pink and the cell will stain blue. That's what the word metachromatic means. The other common thing that's used is tellurite agar. And what happens, the bacteria reduce the tellurites, producing a precipitate of tellurium. And the colonies are seen as black colonies if they're grown on this media, which they wouldn't be, for example, if grown on Loeffler's medium. So both of these tests are used to determine that somebody's got diphtheria. Now, of course, you would not wait to culture. If you suspect somebody's got diphtheria, if you saw a pseudomembrane, for example, you're going to treat. Okay? Okay, well, I got my results 48 hours, but the patient died. Well, that's great. We know that's the diphtheria. You know, I mean, it's not, but you know, these tests are useful because if you've got a case of diphtheria out there and you want to know why do you have a case of diphtheria out there, because you shouldn't have any diphtheria out there. The oxytocin can be tested in vivo and in vitro. It is very difficult to do it in vivo. In this day and age, I don't even, maybe a CDC lab might be able to do it, and I said the word might, but probably nowhere else in the country would. You particularly want to know if there's exotoxin being produced, because that tells you, again, when I said cell-mediated immunity and tuberculosis, and well, it's the same with diphtheria. Say exotoxin, exotoxin, exotoxin. Okay, that's what we think about in diphtheria. Now, diphtheria should not be confused with the so-called diphtheroids, which means looking like diphtheria. There are plenty of Corynebacteria in the normal flora of a normal person. Among them are the Propionibacterium acnes. And so just because somebody grows up something that looks like Corynebacterium diphtheria doesn't mean that they've got diphtheria. It's very, very common to get contamination with Propionibacterium acne. Okay, we'll now discuss the Legionella. I think if I go over 2 minutes, that's okay. At least I got to the point where I can discuss Legionella today. All right, Legionella, gram-negative rod. We're finished now with the Corynebacteria and related organisms, the Mycobacteria, the Nocardia, the actinomyces. This has got nothing to do with those organisms. Why does it confuse? Because, unfortunately, about 30 years or so ago, somebody discovered that one of the species of Legionella that causes Pittsburgh pneumonia is slightly acid-fast. And I have it in my handout for that reason. Most of the Legionella are not acid-fast, and even if they are acid-fast, they've got nothing to do with Mycobacteria. Okay? So they're difficult to stain, but they're gram-negative rods. They're typical of other gram-negative rods. They've got an endotoxin. Anything you care to think about gram-negative rods, these have got it. Okay, there are two types of disease caused by Legionella pneumophila. Legionnaire's disease is the most widely known one, which is a pneumonia which caused so much aggravation and despair when it first came out because people didn't know it was a bacterial infection and they didn't know they could just give erythromycin. As soon as people found out that erythromycin worked for Legionnaire's disease, medical microbiologists and physicians lost interest in Legionella. But parents don't lose interest in it, and air conditioning engineers don't lose interest in it. Because of the fact that it's an organism that's found in the environment. It particularly grows in hot water towers and in air conditioning systems. That's why I say air conditioning engineers. If you talk to an air conditioning engineer, and I don't mean the guy that comes into your house and fixes your air conditioning, I mean experts who build air conditioning systems. If you speak to air conditioning engineers, or they will know about Legionnaire's disease. If you speak to many schools in South Carolina even, you mentioned Legionnaire's disease, the principal probably knows about it, because he may have had a lawsuit against the school. You find Legionella lives particularly the air conditioning system and the water supply, and you can sort of get situations where there's little outbreaks, and they do occur constantly. A small outbreak here and there usually ends up in the newspapers. Legionnaire's disease has not gone away, okay? But it's something that generally, it's very treatable, and it's not treated with the importance that it used to be treated with. Pontiac fever is another disease caused by Legionella pneumophila. Pontiac fever is more of a flu-like disease. It's not a pneumonia. It's milder, it's self-limiting, and you often get muscle pain as you would as in the flu. So you distinguish Legionnaire's disease, the pneumonia, from Pontiac fever, which is the mild disease. They're both caused by Legionella pneumophila. Transmission is in the air, as you might expect. It also infects the water supply. It's not generally spread person to person. And as I said, there can be small epidemics, particularly you see in hospital situations in the immunocompromised. Now, unfortunately, it's very hard to diagnose a case of Legionnaire's disease, and many hospitals don't even try. It doesn't grow on sheep blood agar or any other conventional agar, which is why people had such a hard time even working out that it was a bacterial infection when it was discovered in the middle 70s. The media must contain iron and cysteine, and commonly a thing called charcoal yeast extract agar is used that contains both iron and cysteine. If you know this, and if your clinical microbiology laboratory is using charcoal yeast extract agar, I'm not going to say they're going to pick up every case of Legionnaire's disease, but if they don't use charcoal yeast extract agar, they're sure not going to pick it up. Its isolation from clinical specimens is still poor. Again, when you think about Legionella pneumophila, it's one of the few organisms you think about as an environmental disease like anthrax, although it's not a biological warfare agent. It's found in environmental reservoirs, in pools of stagnant water, particularly water we think about in this situation, hot water towers, air conditioning systems, and it's believed to grow in protozoa. It's a facultative intracellular pathogen in humans, and the reason it grows in protozoa, it grows the same way, presumably as a facultative intracellular pathogen, and that's why it's able to survive in the environment, because it lives inside protozoa. Not always, but that's one of the places it lives. All right, most of the time, there are actually at least 30-odd species of Legionella that have now been discovered. We do use the term Legionella when we're talking about Legionella, just like we're talking about the Enterobacteria ACE. But the major one, the vast bulk of the cases are caused by Legionella pneumophila, and particularly serogroup 1. But the thing you really need to remember is Legionella pneumophila, Legionnaire's disease and Pontiac fever. There's a second disease that's called Pittsburgh pneumonia, which has got nothing to do with Legionnaire's disease and nothing to do with Pontiac fever. That's caused by Legionella mycobacteria, the second major pathogen among the Legionella. You do quite frequently see, now and again, you see other Legionella among these 30-odd species that have been described that can cause disease in the compromised host. And that's the only reason I've mentioned it here, and I certainly do not propose to put all the species names up there. Now, the reason this is a bit odd is because Legionella was discovered in the middle 70s. It wasn't known to exist before that. So we don't have all the sort of metabolic tests that might have been developed 50, 60, 100 years ago for Legionnaire's disease because it wasn't discovered at that point, and people don't tend to work on this sort of thing nowadays. So particularly in this disease, fatty acid profiling is done the same way as I mentioned for the Mycobacteria. The CDC were experts on this. I don't know now that Brenner's probably retired whether they dropped it or not. But anyway, DNA hybridization was commonly used, and 16S RNA sequencing in particular, I'm sure, has become the biggie now. It's difficult to diagnose, to speciate the Legionella. And another test that's been, a commercial test that's available is detection of antigen in urine. I suppose the bottom line is, the punchline is here, there's no good test for Legionella. Okay, if you are successful in culturing it and you find you've got Legionnaire's disease, that's great. But the tests are still not as good as they ought to be. Okay, thank you very much. Sorry I dragged on a few more minutes. If I've got any questions, this is my last lecture. Yeah, go ahead.